Hi guys, so today we're going over chapter 1 of the biochemistry section, and that includes amino acids and protein structure and formation. So to start off this summary chapter 1, we went over all of the different important amino acids and their characteristics and the naming for them, which includes the functional groups that are attached to the amino group. All amino acids have what's known as a carboxyl group, which is this oxygen double bonded to a carbon with a hydroxyl group on it, and a carbon away is bonded a amine group, which is a nitrogen and two oxygens. The rest of the amino acid can be bonded to that beta carbon as well, and it can be any of these R groups, and you have to know all of them. So here I've marked the first main part of the amino acid plus its side functional group in a highlighted color, and the highlighted color coordinates with what the functional group is. So the first green color is nonpolar and non-aromatic side chains, and there's 7 of them, and you have to know the names and the 3-letter abbreviations and the single letter abbreviations. So they're alanine, glycine, isoleucine, leucine, proline, valine, and this one down here, methionine. And one of these, glycine, is special because it is non-chiral, and that means that it doesn't have chirality. They explain that later. And all of them except for cysteine, which is a different one, has the S configuration. Now, the importance of the side chains is that they are what make each amino acid functionally different. And we're going to go over some of the uses later. But the next group, the pink ones, are the aromatic side chains. And there's 3 of them: phenylalanine, tryptophan, and tyrosine. And those are relatively nonpolar. Well, phenylalanine is actually the one that's nonpolar. Tyrosine, since there's a hydroxyl group, it is actually semipolar. And next we have the relatively negatively charged side chains, and that's going to be these acidic amino acids, and they can be formed in their protonated or deprotonated ones, which means there's an oxygen on the end which can be written with or without the hydrogen. So these are aspartic acid and glutamic acid, and their non-protonated form is aspartate and glutamate. Later we're going to go over at what number of pH these guys exist in and what level of acidity. Next we have the positively charged side chains, arginine and histidine and lysine. And this didn't fit, so it's over here. But they are side chains that have amino acids on the end, which can be protonated additionally. And in arginine, there are 3 nitrogen atoms in the side chain, which means the positive charge is delocalized over the 3 nitrogen atoms. It means there's some resonance in there. And in histidine, it's a little bit more complicated since it's an aromatic ring. And on a side note, the ring is called an imidazole. And the way that it retains a positive charge is that the pKa, which I'm going to explain in a minute, is lower than 7.4. So at what our physiological pKa, or our physiological pH, one nitrogen is protonated and the other isn't. And under more acidic conditions, the second one can actually be protonated and it becomes positively charged. Lastly, we have these polar side chains. We have serine, threonine, cysteine, asparagine, and glutamine. And they can participate in hydrogen bonding, which means they can possibly be used in beta sheets or alpha spirals. And there are also a few of them, asparagine and glutamine, which have ketones in their side chain, which means they can participate in carboxylic acid derivatives and synthesis. So that should be known also. So those are the amino acids that I'll have to know in detail, and you're supposed to know every piece of it. So those are them. The next part of the chapter talked about acid-based chemistry, which is kind of complicated, but I'm going to go over the summary and the key points. So there's some key points here. If the pH is less than the pKa of the amino acid, then it was most likely to be protonated. And if the pH is greater than the pKa, then it's deprotonated. And if it's around the same, then it's neutral. And the neutral amino acid is called a Zwitter ion. So, as mentioned, every amino acid has a carboxyl group and an amine group, which means there will always be two pKa's for each amino acid, and some of them have a pKa for a third part of it for the ionizable R group. It's important to remember that the pKa for the hydroxyl group in the carboxylic acid is usually around 2. And the second part is the pKa for this amino group is a 9, in between 9 and 10. This is important because that means at anything below the pKa of this group means that both of them will be protonated. So at a pH of 1, that means that there will be a hydrogen here and a hydrogen here. But if we increase the pH to above the pKa of this carboxyl group, that means that this oxygen here will be deprotonated. But as long as we stay below the pKa of this one, then this will stay protonated. So if we take an example in the body, the stomach acid pH is around 1 or 2, which means that the whole amino acid will be protonated always. But if we go into the bloodstream or something where the pH is more 7.8 or 7.4, then we are above the pKa of this one, so we are deprotonated, the carboxyl group, but we still have the proton in this amine group. This gives the molecule a neutral charge, and that's why we call it a Zwitter ion, for the German word Zwitter or hybrid. Another thing that was mentioned in this chapter was the isoelectric point, or the pi of the amino acid. And that is the point at which the molecule is electrically neutral. And unfortunately, there's 3 different equations to figure that out, depending on whether the amino acid is neutral, acidic, or basic. And if it's a neutral amino acid, the formula is the pKa of the amino group plus the pKa of the carboxyl group divided by 2. So it's basically an average. And for acidic groups, it's the R group plus the carboxyl group divided by 2 because it has a lower pKa usually than the amine group. Conversely, if it's a basic amino acid, then there's the formula, the amino group, the pKa of the amino group plus the pKa of the R group divided by 2, because it becomes neutral only when it loses the proton from its main amino group, which happens around a pH of 9. The main point of this is that amino acids with acidic side chains have low pIs, and those with basic side chains have high ones. The rest of the chapter covered the structure and formation of proteins, which I know pretty well. They covered the primary, secondary, tertiary, and quaternary structures of proteins, and the main points was that those are formed through, well, the primary structure is formed through covalent bonds between the amino acids. The secondary and tertiary structures are formed through hydrogen bonding, and the quaternary structure is formed through many different things: ionic bonds, hydrogen bonds, and other types of folding. There was a highlighted point that in cysteine molecules bond to form cysteine, they can cross bond using the sulfur in their side chain to form disulfide bonds, and those create loops in the protein chain. There was also mention of the hydrophobicity of proteins, which explains how hydrophobic molecules, amino acids, are going to be on the inside of the protein folding, and hydrophilic molecules are going to be on the outside of the structure. Finally, the chapter talks about conjugated proteins using prosthetic groups, which are different things attached through covalent bonding, such as organic molecules, vitamins, metal ions, and even sometimes lipid groups, carbohydrate groups, and nucleic acids. So that's the end of chapter 1. Next week I'm going to go over chapter 2, and tomorrow I'm going to go over biology, chapter 1. So thanks for watching, and I'll see you tomorrow, I guess.